in this section, I'm going to talk about advanced search. I'm going to show you how to build your queries to find the proteins that you are interested in. I will show you what to do in the case that you get really long list of results, what clustering strategies do we provide, and finally the 1D-3D, a really useful tool to explore sequence annotations and map them on the 3D structure of macromolecules. Okay, so first, what is advanced search? So structural data comes with a large collection of metadata. For example, you could be interested in finding these assemblies that has a molecular weight greater than a certain threshold. You could be interested in finding structures that contains a protein with a chain of a particular length, or you could be interested in finding high-resolution crystallographic structures. So advanced search will allow to combine all these different attributes, but even more because we also integrate many external resources. So for example, RCSB PDB data is annotated with protein classification domain databases, as for example, CATH, SCOP, PFAM. We integrate UniProt annotations, gene ontology terms. You could be interested in finding all the structures related to a particular cell process or a metabolic pathway. And also, we provide different types of scientific searches. So for example, sequence identity, finding those structures containing a sequence that is similar to my sequence, or a structure similarity search. So we provide a tool that is the query builder that will allow you to combine all different types of searches into a single query. I will show you also, once you get the results, what operation can you do with them. So we will talk about the search refinement tool. No? This is going to be a really useful utility once you have your result to add new conditions to your initial query. I will talk about what different ranking strategies to sort your results are provided. And finally, in those cases where you have a list of thousands and thousands of results that cannot be inspected manually, we also provide different cluster strategies to try to reduce redundancy and method to explore groups of proteins, not these proteins that has been clustered together and under a certain condition. Okay, so the advanced search query builder. So as I said, the advanced query builder, what does is merging all these different 8 services into a single query. I'm not going to go into deep detail, but I would like to just to give a brief description of each of them. So we have a full text service. This is a free text, a Google-like. So type your text and we will do our best. We have a structure attributes. As I said, a structure information comes with a long collection of metadata. You would be able to build your Boolean queries, adding conditions on molecular weight, sequence length, and also including these external resources, CATH domain, a particular pathway identifier. So similar to structure attributes, we also provide the chemical attribute search. So in that case, it's attributes related with the chemical, the small ligands that can be found within the structures. You could be interested in finding, give me all the structures that contains, for example, ATP. Sequence similarity, I am completely sure that most of you have used the BLAST search. No? So in this case, we are not using BLAST. It's a much optimized algorithm called MMSEX. And what it will do is just finding similar sequences to a given one. The structure motif search, I don't want to go into detail what a regular expression is, but the sequence motif I am talking about. The sequence motif will allow you to find these local sequence patterns. Let's say that I am interested in finding all the structures that contains a sequence with a methionine, followed by a methionine, any other residues, and finally a glycine. No? So the sequence motif search will allow you to define this local sequence pattern and find those sequences. A structure similarity, so this is a search based on 3-dimensional shape recognition. So it will search for structures similar in the 3-dimensional conformation to a given one. Finally, we have two more. A structure motif is similar to the sequence motif, but in that case, the motifs are defined as geometric patterns. So three-dimensional local pattern defined by the selected residues. And finally, the chemical similarity that will allow you to find those structures containing ligands similar to a given chemical smile, or also we provide a sketching tool to input your two-dimensional the two-dimensional graph of your molecule. So advanced search also allows to configure a bit deeper what you want to get. You can see here on the bottom different options. So let's see what these options can be used for. First of all, the return type. So this will allow you to choose what type of objects do you want to get. No, structures, you will get experimental PDB or computational structure model entries. Polymer entities will return all the distinct chains, proteins or nucleic acids that are found in the different structures. Assembly will return the macromolecular quaternary structure, sometimes also called the biological unit. So in many cases, the structure will not be the conformation of the native complex, but to build the complex, some transformations are needed to be applied to the chains contained in the structures. And in other cases, one structure will contain different assemblies as a combination of a subset of chains. Non-polymer entities will return all the chemical small ligands that are found in the different structures. And finally, the molecular definition, so the chemical components that compose the different macromolecules of the structure. Now, in that case, in most situations, you will be able to find the 20 natural amino acids, non-standard amino acids, nucleic acids, and other chemicals that are found, small ligands that are found in the structure. Okay, I have been talking about what to do when we get a really long collection of results that cannot be explored manually. So for that, we provide two different clusters strategies. One's based on sequence identity that will be clustering your results based on the sequence identity threshold provided, and the other based on uniprotaxation aggregation. So in that case, groups, what will happen is that all the members of the groups, all the proteins, will share the same uniprotaxation. And finally, in many situations, we will not be interested in exploring single proteins, but family of proteins that are related together with the given condition. So for that reason, we provide also these two options. Once we have grouped the results, we can do, okay, so I am only interested in having a non-redundant set of results. So I am only interested in displaying one representative, one protein for each single group. Or in some other situation, no, I am interested in knowing what is going on. I need an overview, not only of my protein of interest, but also on the related proteins. So you will be able to display the results as groups of proteins and not as single items. And finally, we have here the switch to include or exclude computational models. Okay, so okay, here we are. So this is how a search result page would look like. Here, what is most important, remember, the navy blue color Elden-Meyer flask means this is experimental information, and the cyan color screen icon means is computed structure models. So we get here different items, as I said, by default, so we can use different scoring strategies to sort the results. By default, the order depends on the search score, and this depends on the type of services that have been used. So for example, if we are searching by sequence similarity, results will be sorted by sequence identity scores. but we provide on the top right side of the result list, we have this drop-down sort by that can be used to select the different ranking strategies. One might be interesting. No, I want to sort that by release date or by resolution. I want to see first X-ray crystallography with better resolution and then the others, or I could be interested in computational models and sort them by the local scores, etc. So many other different ranking strategies. Also on the left side, what you can see here is what we call the refinement tool. So this will allow you to filter the results based on the attribute value selected. No? So for example, if we focus on the first two different checkboxes, it says a structure determination methodology, experimental 2, computational 1. That means that in my result list, there are 2 experimental items and 1 computational. I could click on experimental and only obtain the results, including the experimental condition. So I would remove from there the computational structure models. Also, I was talking about grouping. Of course, that this is not a good example because it contains only three items and can be inspected manually. However, what could I do if I had 3,000? So we could use clustering. So in this case, these three structures contains only a single protein each of them, and all the proteins is the same, at least from the UniProt point of view. They share the same UniProt accession. So what would happen if I group them by UniProt? That my result list will become a single item. On the left side, I am seeing what I will get if I choose to display the representative. So I have a single group and I'm displaying a structure for each single group. So one group, one structure. And at the right side, all three structures say the same UniProt accession. So all of them has been grouped together into a single group. Okay, so as I said, groups are explorable. Similar to the structure summary page, we also provide the group summary page. And group summary pages would look something like that. So for example, this is a group summary page based on UniProt. So on the left side, you will see a carousel that will allow you to explore the different members of the group. Here you can see member 1 of 3. Remember, this is a group of size 3. We provide the UniProt name, UniProt IDs, here we have the UniProt description of the protein, and we have now different histograms. So these histograms, what display are the distribution of the different group members' attributes, because now I cannot display a release date because this is a group, I have 3 proteins, but what I can do is display a histogram of the release dates. I will show you this when we go to the live demo. And on the right side, what we can see is what we call the group sequence page. So in that case, as it is a UniProt-based group, what we can see are on the top of the protein feature view, we can see the UniProt annotations, and at the bottom, the multiple sequence alignment of the group members. So those that contains this color code scheme, it's because they are computed structure models, and we are showing in these orange to deep blue colors the local score. So the blue regions are those that has a good local score, a good prediction, and those in yellow orange are those that are not that good or can be considered as unobserved regions. So what I think this view can be quite useful because it gives you an overview of the relationship between the local scores and the alignment of the experimental structures. Okay, the most important thing, how to go to advanced search. So each of the pages contains this link that is on the top of the page, just under the main search tool. So you could see the link advanced search. So clicking there, it will navigate to our advanced search page. However, what I find most interesting are those links that can be found in the structure summary pages. So our structure summary pages are really well connected with the other residues. So for example, when I go to the macromolecular section, I can go to the advanced search and find sequence other structures that contain sequence similar to the one that I am interested. The same with three-dimensional structure search. No? Also, we provide, for example, search by UniProt. Give me all the structures that contains a protein that shares this UniProt here. And we have many more links. No, we have, for example, organism. We'll also go to the advanced search and search by all Homo sapiens structures, in that case, gene name. And as we provide links to the advanced search, we also provide links that goes straight forward to the group summary pages. So in that case, all these group summary pages, what has in common is that all of them contains this protein here, these computed structure models. Okay, so finally, I would like to also show what is the 1D-3D view. So this view, what will provide you is an easy way to explore positional features from the 1D on the 3D. To access that, you can do it from any structure summary page, always in the carousel, in the section where it says 3D view, you always will find this 1D-3D view. Okay, so let's go to see all these tools that I have been showing on our website. Okay, so first of all, the main link, advanced search. I click here, I go to our advanced search page. This is our query builder. As I said, this can be used to merge all different type of searches. For example, I could be interested in a sequence similarity search. I can go here in this text box, just type my sequence and go for search. Or here we have our free text. Type whatever you want, and it will run this free text search for you. Okay, so let's now inspect the different links that I was explaining before. So for example, we have here the structure similarity search proteins by UniProt. So in this example, what I would like to do is search other structures that contains a chain that is similar to 30% to our protein, in that case, the NOVA2 protein. So when I click here, we can see that it browses to our advanced search. Okay, before going into the results, let's see what has happened with the query builder. So as we can see, clicking there, what has done is fill the sequence similarity search with the actual sequence of our NOVA2 protein. And what we are seeing here are, remember, science screen, computed structure models, and Eldenmeyer flask, these navy blue experimental structures. Okay, as I said, the default order depends on the type of search service that has been used. In this case, it was a sequence identity. So if we check what are the sequence identity values, 100%, 98%, 78%, so it decreases because this is the default search for the service. In case that you use multiple services, what the advanced search will do is merging all these scores into a single one. As I said, we have all these ranking strategies. For example, look, I am not that interested in computed models, so I would rather see experimental structure first. So we have here a priority ranking, experimental first, and what now we will get in the first page is only experimental structures. And to find the computer structure models, we have to go to the last pages. Okay, so now refinement tool. As I said, we can now refine this initial query, no, that was based on sequence identity. So in my case, I could be, look, my project is about Homo sapiens, so I am going to be interested in only Homo sapiens protein. So we can just click there and refresh our search, and now what we will get is just the subset of our initial search, only containing, as you can see here, organism, Homo sapiens, organism, Homo sapiens, organism, Homo sapiens. However, refinement tool is a very nice utility because it is quite easy, quite fast, and being able to see the number of proteins, the number of structures that you will get is really useful. But not everything can be done. So imagine that I want to compare now good high resolution structures with computed models. So one could be tempted to go, okay, so let's choose all these ranges of resolution, let's say up to 3 Armstrong, and run the query. So pay attention here. I have 24 experimental, 7 computed models, and run this search. And let's see what happens here. Oops, I have lost my computed models. And this is because the computed structure models, they do not contain resolution information. It has no sense in that context. So as they don't contain, they do not appear here. So for that, to write these more complex queries, we need to use the structure attribute search. Look what has been happening here. I have not talked about that, but each time that we are adding condition from the refinement panel, the advanced search query builder, what does is include those attributes as part of the structure attribute search. Okay, let's go back when we didn't include this refinement resolution, and let's see how we could do that. Another really cool utility is the query history that will allow you to navigate among all the queries that you have done. So it's quite easy now to go backwards. And here we are again, only with my sequence identity condition and my Homo sapiens condition. So what I was saying, I want to include computed models and experimental structures with a resolution lower than 3. So what I can do is, okay, let's add, sorry about that, this is not going to be enough. Let's add a new subquery where I will say determination methodology is computational. Let's add a new attribute, or refinement resolution lower than 3 Armstrongs. Let's run the query. And here we are. Now we have recovered these 7 computed structure models, and all these experimental, I know that are coming from X-ray diffraction with resolution lower than 3 Armstrong, 194, 2.3, 2.4, 2, etc. Okay, let's talk. I know that this is not the best example because one could say, oh, these are only 25 items. I can explore them manually, but just for the sake of explaining what is the grouping tool, let's apply grouping here. And let's say, okay, this list is too large. I want to see only those proteins that has a different uniprot accession. So because this is what I'm interested in, I'm not interested in that large amount of result. I want to remove some redundancy here. So I can run the search now grouped by uniprot accession, displaying a representative. So now I am getting only 11 search items, and this is because those are the unique uniprot accessions that are found in my initial search. So this is a representative for a group. So behind these different items, there are other proteins, and only the representative is displayed here. If we were interested in the situation where you are not that interested in a single protein, but exploring or studying a family of proteins, what you could do is display results by groups. of course, that we will get the same number of items, but in that case, each of these items represent a set of proteins, a group of proteins. And as I showed you before in the slices, let's see how the group summary page looked like.
Excuse me, Joan, 5 more minutes.
Yeah, sure. So here we are in the group summary page. As I see, as I explained, the carousel displaying the different group members. We have these different histograms, and it's quite useful. Histograms behaves quite similar to the refinement tool. So when I am here, I could be interested in, so I am only interested in X-ray diffraction members of this group. So I can click here, and now all histograms will be updated, and the blue portion will be reflecting what is the distribution under my condition, in that case, X-ray diffraction, and the gray are the rest of the protein. So look, for example, here, when I have selected the X-ray diffraction member, the computational has disappeared to 0. Okay, so let's go to inspect the full group, and let's go to the group sequence page. Yeah, as I said, those are annotations coming from UniProt, modified residue, some domain information, and here we have the group multiple sequence alignment. So if I zoom in, I could go to sequence level and really check that this is a real alignment. No, so and what I think is most important or more useful here is that I have the relationship between those regions that can be, let's say, trusted from the computed structure models. So for example, this region here, and I have also the experimental structures that covers those regions. Okay, so finally, really fast, because I am running out of time, let's go to see the 1D-3D view. As I said, in any summary page, 3D view, you will find the 1D-3D view here, and it will open this really cool view where you could see all different types of positional features. We have the secondary structure, we have predicted disorder, we have here the PLDDT values, and of course that this is dynamic. No, I can click on one feature and it will be highlighted on the 3D structure in the viewer. No, so I think in some situations this can be really useful because it allows you to have an overview of all the annotations. So for example, here we can see that coming from a completely independent method of prediction, that the regions that has not been well predicted or cannot be predicted with the computed method also matches with what other methods predicts that disorder is happening. So yeah, this was my presentation. So thank you very much for your attention, and I will be really happy to answer any question. For the group summary, no, not really. We don't include that that type of information. However, if you think this can be useful, of course that we are open to listen any opinions, because we didn't think about that, and maybe we were missing something that will enrich our group summary pages. So we'll be quite happy to hear your opinion.
Thank you. No, and we recommend that you use something like the DALI server to do those searches. We again, as Joan indicated, if there is a strong interest among RCSB.org users for that functionality, please make it known to us so that we can evaluate that alongside other opportunities for doing improvements.
I think a flavor of that may actually be possible, and this will come up in Sebastian's talk later,
Okay.
where you can upload files and do some level.
You can certainly upload files and do binary comparisons, but doing a PDB-wide search, I think is
Motif
still,
search are possible.
yeah, I think it's still the purview of something like DALI.